Endocrinology. The concept and methods generally are going to be endocrinology and hormones, chemical messengers, kinds of chemicals messengers, neuroendocrinology, anatomy, cells and organelles, secretion, chemistry, amounts, cyclic nucleotides and genes, molecular biology related, methods of anatomy and histology related, methods of chemistry, methods of physiology, related statistics, rhythms, species, specificity, hierarchies, set point regulations, negative feedback regulation, target receptors, mechanisms of action, semasiology, story, organizing endocrinology. We're also going to see the tables, and I'm going to ask you to take a look at the tables and the drawings your book, the book you choose, has given you, so you can take a look at it and put things together. We'll see the receptors, mechanisms of action, and prostaglandins. The receptors, the affinity, the receptor structure, regulation of receptors, receptor-mediated endocytosis, cooperativity, spare receptor, social amoeba, second messenger hypothesis, cyclic AMP, phosphodiesterase, cascade of reactions, transduction, cyclic AMP, assays, analogs and blockers, cyclic GMP, calcium and calmodulin, kinases, phosphosinositides, hormone action and vision, prostaglandins, cytoplasmic receptor for steroids and thyroid hormones, criteria for distinguishing between cyclic AMP and steroid mechanism of action. Sequence of endocrine function disease involving receptors and mechanisms of actions. We priorly posted a program, right, to nurture the community's curiosity, and we have posted it with some drawings, and we wrote it by hand, but we are going to just develop a little bit deeper for everybody, so get to know a little bit more about their human body with the same idea. We are saying that because the evolution has produced fine systems, in this case two, which work together to perform something we call communication function in multicellular organisms, where the first is the nervous system, and the second system, and the one that is the topic of this book, is the endocrine system, right, which is the book that I use, or the book that you use, right, the matter of what we are talking, right. And we are thinking and saying that acts a little bit more slowly. The endocrinology, defined by Webster's, is a science which studies the endocrine glands and the internal secretions of the body, which is derived from Greek endo, which means within, crine, to separate. Biologist thinks sometimes and gives a thought that the nervous system is some sort like a writing system, and where signals flash from cell to cell at a very high speed. And appreciating the interaction in between nervous system and endocrine system provides a view of thinking that these two systems functioning together to use messengers, in this case chemical messengers, which are signaling, right? And therefore it explains how the student might be able to discover, right, about the endocrine glands, um, and what probably mean by internal secretions. Endocrinologists sometimes talk about endocrine glands, right? Which are some sort of like a ductless glands, which are secreting products into the bloodstream. Classically, understanding endocrine glands such as pituitary gland, thyroid, parathyroid, adrenal, the gonads, the pancreas. Organs which are glands with clear boundaries visible to the eye, and of most vertebrates. Understanding vertebrates also other than human beings, mammals, birds, reptiles, amphibians, and fish. And some cells of some other kind of tissues, such as thymus, pineal, hypothalamus, which are messengers secreting chemical substances that might be considered to be endocrine. Therefore, these chemicals messengers travel in fluids other than blood, such as cerebrospinal fluid, etc. The chemical messengers, classical hormones, come from endocrine glands. The word hormone is understood as specific chemical product of an organ, of a certain cell of an organ, which is transported by blood or other body fluids with a specific regulatory effect on the cell remote from its origins. And the hormones are all molecules. The word hormone comes from a Greek word hormaein, meaning to excite or to arouse. Probably they are called by their functions as the chemical messengers. Chemical messengers is a molecule which has information signaling from one location in the organisms of the human body to another site in the same body. Chemical messengers are viewed as travelers through fluids. These messengers are not chemical. Classical hormones are called hormones, but this practice is probably known by classical oriented endocrinologists. The communication by chemical messengers are of four types. It says over here the endocrine communication using the circulating body fluids, the neural communication via synapses, the paracrine communication when the messengers do not enter the bloodstream but act on other cells, and the fourth one, the autocrine communication when the messenger act on the cell from which were secreted, possibly for a chemical compound to have autocrine, paracrine, and endocrine effects. Is given an example of testosterone's concentration in cells of the testes affecting the rate of testosterone synthesis, autocrine effect, and diffusion to other testis cells affects spermatogenesis, paracrine effect is called, and testosterone circulating through the body affects physiology and behaviors. And this is called the effect endocrine. The kinds of chemical messengers. It is known that the hormones are chemical messengers. Some scientists might not view all chemical messengers as hormones. The classical hormones secreted in bloodstream by ductless glands are subset of chemical messengers. Specific hormones names are thyroid hormones, estrogens, and prolactin. The nervous system uses chemicals for communication, molecules, and its functions. A neuron, which is considered the nerve cell, communicates with a neuron of another cell by secreting chemicals through a synapse or cleft. A synapse is a space in two neurons. The chemicals liberated at the nerve endings crossing the synapses are called neurotransmitters or neurohumors, traveling one cell to the next without using bloodstream. Communicating these two cells across a synaptic cleft exists only one of the ways that nerve cells work. Some neurotransmitters are acetylcholine and norepinephrine. Some other chemicals which we are talking about are ions and small molecules which pass between adjacent cells, where membrane cells are in contact with gap junction. Neural signals transmit directly cell to cell without chemical messengers by gap junctions. And this passage is not considered to be endocrine. Molecules are secreted, some of them, by nerve cells which are specialized into the bloodstream. The nerve cells which do this work are called neurosecretory cells. The hormones secreted in the blood by neurosecretory cells are called neurohormones. All cells make molecules such as urea, histamine, carbon dioxide, wound hormones, embryonic inductive substances, etc., etc. Some of these molecules are found in the bloodstreams, influencing this way other cells, and they can be considered chemical messengers. Parahormones is used by some endocrinologists, saying that the probes of glands or chemical messengers that are not established as endocrine glands, right? Plants also have chemical messengers. They don't have a circulating bloodstream. Plants' messenger molecules cannot meet the classic definition of hormones. The phytohormones means plant hormones is used to mention chemical messengers in plants. And there are used some examples such as phytohormones, which are auxins and gibberellins. Something that is very interesting in this group of chemical messengers is that is different from all the previously described chemical messengers carrying information within organisms, which are messengers, chemical messenger between organisms. And these are called pheromones, which are messengers transmitted through the external environment, controlling interactions between individuals. And these are species specific. Pheromones are sex attractant of insects and the chemical signs found in mouse urine. The neuroendocrinology is used to the endocrinology of those glands in which neurosecretion plays a large role, and the hormones are neurotransmitters. The neuroendocrinology includes endocrinology of the brain, pituitary gland, adrenal medulla, and pineal gland. The relationship between a hormone, a neurotransmitter, and a neurohormone can be a little bit confusing. To be clear, the classic hormones are secreted by endocrine cells in the bloodstream. Neurotransmitters cross a synapse between two cells, and neurohormones are secreted into the blood by neurosecretory cells, which are called nerve cells or neurons. A region where the neurohormones can be released or stored is called neurohemal organ in vertebrates. It says over it mentions in this book the median eminence of the hypothalamus and the posterior pituitary gland are the only neurohemal organs. There are other other neurohemo organs in non-mammalian species. The caudal urophysis of fish, the sinus gland of crustaceans, and the corpus alatum and corpus cardiacum of insects. The anatomy of the endocrine glands are readily located visibly in the head, torso, and scrotum. The pineal gland lies in the anatomical center of the brain in humans. Sometimes it was known as the primary location, which means the pineal gland was the location of the soul, right? Just to be said by Descartes. Some mammals, such as birds and lower vertebrates, the pineal is located towards the top of the brain and might even lie just underneath the skull. Below, and finally we say the pituitary gland is below the brain in all vertebrate species. The thyroid gland is found at the neck of humans, and the parathyroid are on the thyroid. But in other species, the thyroid is in the chest and the parathyroids are separate. In humans, the thymus is found in the upper chest and it is large early in development. The pancreas nests among and overlays the loops of the intestines. The adrenal glands are found very close to the kidneys. The ovaries and testes are called the gonads. The ovaries are in the abdomen, and the testes might be either in the abdomen or in the scrotum. The cells and organelles. The cells of the endocrine gland have organelles which are used in the synthesis and the secretion of hormones. The nucleus of each cell contains chromosomes made up of molecules of nucleic acid used as blueprint to make hormones and enzymes. In the cytoplasm outside the nucleus are organelles such as Endoplasmic Reticulum, Golgi apparatus, mitochondria, phagolysosomes, which are very important for the synthesis of all these hormones. The Golgi apparatus is very close to the nucleus and has 6 or so membrane-bound stacked sacs called cisternae. Golgi apparatus processes proteins, modifying as well sugars of glycoproteins. Endoplasmic reticulum is a series of tubules, membrane walls of tubules, either rough granular with ribosomes or agranular which is smooth. Structural types of the hormones are synthesized by an endocrine cell related to the detailed anatomical appearance of the endoplasmic reticulum. The polypeptides synthesized by the ribosomes. Steroids are synthesized by the smooth endoplasmic reticulum. The mitochondria, organelles responsible for oxidative phosphorylation, the mitochondria energy stores the molecule of ATP, adenosine triphosphate, and is by the size of some steps in the synthesis of steroids. The lysosomes, organelles, which contain digestive enzymes, which has the function of cell's digestive system, the thyroid gland, the phagolysosome, involved in the process of releasing the thyroid hormones from their storage in thyroglobulin. The cytoplasm contains other organelles such as vesicles, lipid droplets, vesicles containing secretory granules. And they are important to hormone synthesis, storage, and secretion. Cytoplasm also has centrioles, microtubules, and microfilaments, forming dynamic skeleton for cells and the use of cell division and animal movement. The microtubules form tracks for the movement of secretory granules to cell membrane. In times, the cytoskeleton is known to be affected by hormones. The cell membrane, called plasma membrane, particularly important for hormones because they have to pass through the cell membrane to get out from the cell and go into circulation to deliver their message. The membranes are made up of proteins, cholesterol, and phospholipids, which have a head and two tails because the phospholipids have soluble polar hydrophilic and the phosphate heads and insoluble non-polar hydrophobic tails. The heads contact the aqueous external fluids and cytoplasm, and the heads sandwich the tail between them, embedding proteins like nuggets at the membrane side on the inner or outer surface or all the way through the membranes. The proteins provide the structures to the membrane, and they work as pumps, actively transporting ions through the membrane, forming ions channels. They work as enzymes and act as receptors. The secretion, endocrine cells, we know, secrete their hormones into the external space, and then the hormones enter the bloodstream walls of capillaries of most endocrine glands, facilitating movement of large molecules in the brain and also in the muscles. The fenestrations are absent. The absence of fenestrated capillaries in brain and muscle contributes to the blood-brain barrier, which protects the brain from outside chemical influences. The peptide hormones are known secreted in the manner of proteins and amines. Some sort like to the secretion process, which are made by cells that secrete proteins or enzymes, and by nerve cells that secrete biogenic amine neurohumors. The proteins and their precursors, which are given the name of pre-hormones or prohormones, are synthesized at the endoplasmic reticulum, which we already mentioned, that organelle in the cytoplasm of the cell. And this protein passed through another organelle, the Golgi apparatus, where they are packed into secretory granules in vesicles. And the hormone probably could be stored and later processed in the vesicles. Or the hormones might be secreted from cells immediately. This secretion involves the traveling of a dense core vesicle through the cytoplasm of the cell to the membrane cell, where the vesicle releases its contents, which are hormones or granules, into the external space by a process called exocytosis. Secreting amine hormones is some sort like to that of protein hormones. In the adrenal medulla, tyrosine converts to dopamine in the cytoplasm. Dopamine concentrated into secretory granules where enzymes convert it to epinephrine and norepinephrine, and therefore the hormones subsequently are secreted by exocytosis, and where the membrane is conserved by recycling. The steroid hormones, in another way, are synthesized from precursors cholesterol and lipid droplets, a shuttling affair that takes place in the cytoplasm, where the smooth endoplasmic reticulum and mitochondria of steroidal cells transmit to the bloodstream is simply because the lipid-soluble steroids can pass rapidly through the cell membrane. The rate of steroid secretion is usually a function of the rate of steroid synthesis. The proteinaceous molecules in the bloodstream act as carriers for molecules of steroid hormones. The chemistry. Usually most of the hormones fall into different categories of chemicals. The polypeptides, glycoproteins, steroids, and amines. And many of the hormones are proteins. Like we were saying, proteins are assembled from small molecules called amino acids, right? Where residues are bonded together by peptide bonds to make proteins. These from amino acids are peptides, polypeptides, called or proteins. Some of the hormones containing amino acids and sugars are classified as glycoproteins. Peptides having 2 and 10 amino acid residues. Polypeptides have 10 to 99 amino acid residues, and proteins have more than 100 amino acid residues. And therefore, practicing this distinction, we can be able to know and to refer about what it belongs to each one of them. Hormones that are structurally similar to amino acids are amine hormones. Iodinated amine hormones from the thyroid also have iodine in their structures. Hormones made by testes and ovaries and adrenal glands are steroids. Steroids have a structure, a very distinctive structure with 4 rings which are connected and side chains. A group of chemical messenger is derived from fatty acid and they are called prostaglandins. It seems at first that there is an overwhelming names of hormones, right? About learning how to identify each one of them, we can simplify everything by grouping and where they belong. Because they have a family and because they have a structural similarities. By this time, I would like you to take a look in your book, right? All the figures that there are, right? Starting from the interaction of the chemical messengers at the nerve system, right? And also the work of the cell, right? With all the sites that we mentioned, right? And let us take a look at the table and how the peptides are assembling, right? And that's the way we will be able to find, right? And let us take a look at the diagram and the figures given in your book and how the secretory glands, right? And how the secretion event takes place, right? So that way you can know the amounts and how is released from the invagination and how is sent to the exterior or to the interior, right? It depends how is developed. The cyclic nucleotides and genes, some compounds are small molecules chemically related on bases that form DNAs or RNAs. And they have a function intracellular. Second messengers are called the cyclic nucleotides. The cyclic adenosine monophosphate is one of the cyclic nucleotides and is the AMP. And genetics becomes an important field to endocrinology because the genes are known to be responsible for the structures of protein, hormones, enzymes that synthesize hormones and hormone receptors. And because the genes, the precisely arranged sequences of deoxyribonucleic acid in the nucleus controlling hormones produce, providing genetic code for the synthesis of hormones that are made up of amino acids such as peptide, polypeptide, and protein hormones, which they provide genetic code for the synthesis of enzymes and synthesizing the other hormones such as amine, prostaglandins, steroids, so and so. And by themselves are not proteins. Genes also are important because they code for the receptor protein that are in target cells. expressing the receptor genes, regulating by the amount of hormones, right? The molecular biology related is the biochemistry and genetics of the nucleic acids such as DNA and RNA, which is the genetic material containing the blueprints which will process the peptide hormones and for the enzymes involving in the information of the hormones. There are not peptides. And the advances of this science, molecular biology, provides a technical assistance because they provide enormous potential to, application into the endocrine system. These techniques can be used to synthesize peptide hormones in great amount and large quantities. It says in your book that peptide hormones were extracted in minuscule amounts from animals or people. And molecular biology can also be used to further explain how hormones act, right? All these techniques provide the means by detect genetic bases and diseases and probably provide possibly genetic repairs of abnormalities in the synthesis of hormones which are associated with substances which are called receptors, synthesizing enzymes, etc. Hormones that are mentioned constantly are peptides and polypeptides. The amino acid sequences of the precursors of these proteins are specified by the sequence of bases in deoxyribonucleic acid, which is a DNA, which is genetic material. And hormones like this, there are small molecules, are synthesized from nutrients by enzymes which are activated, right, by those nutrients. Hormones, right, like the hormones, the enzymes are proteins where the amino acid sequences becomes specified by the blueprint of bases in DNA. And these instructions makes hormones that are in the genes, and therefore the ability to make these hormones and the exact sequence of amino acids and these hormones is inherited. In order to find a specific gene or to make its peptide product, right? In this case, we are talking about hormones, right? A structure of a peptide hormone must be first worked out, right? Endocrinologist, right? Following the sequence of amino acid of most peptide hormones has already been accomplished. The nucleotide sequence of the DNA for the hormone can be deducted, right, or found from the amino acid sequence of the protein, and therefore can be produced, right? The DNA can be produced by chemical synthesis, can become from chromosomal DNA or produced by reverse transcription. Also is found the recombinant methods, which is the promised mass production of polypeptide hormones, which right, we mentioned before that it was given in minuscule quantities, right? obtained from living organisms or dead, right? Their blood, their urine, right, were used to see what kind of hormones were possibly obtained from it. And therefore, it means that all these hormones are available for clinical use. The first hormone who was made using the recombinant techniques was the somatostatin. And you can see right in your tables, right? Take a look at your tables, all the substances, right, that there are under the peptides, the polypeptides, the proteins, under the glycoproteins, under the steroids, under the amines, right? How the hormones are classified into families by structural similarities, right? How the hypothalamic hormones, right? Neurohormones, right? The hypophysiotropic neurohormones, right? The GI hormones and related molecules, the melanotropins, right? The insulin-like hormones, the prolactin-like hormones, the iodinated amines, the endolamines, the steroids, the glycoprotein hormones, the catecholamines, the other hormones, right? To get a look, if your book has the molecular configuration of each one of them, right? It's always nice to know because they are classified in order to make everything easier, right? You find the classification by the biochemical categories and by the structural similarities, and therefore everything becomes very easy. And there also the amounts found present in minute amounts, right? Take a look in your book, right? The diagram where it shows the flow of information from genes, DNA in the nucleus to the assembly of proteins at the ribosome into the cytoplasm, right? It's a very interesting way and methods, right? This is like we were talking, right, before, and which is something very nice. The insulin. Take a look at the chart and the figure, the production of insulin by the use of bacteria, right? Is the insulin A chain and the insulin B chain, right? By the oxidation in the air, the variation of both of them, right? And over here, how the use of E. coli bacteria with plasma DNA containing instructions for making insulin A chain, and how the E. coli bacteria with plasmid DNA containing instructions for making insulin B chain. The biological variability, which is used in statistics, right? Which is used in endocrinology, right? You can find the table, right, and where the N-acetyltransferase activity in the pineal gland, right? This is an enzyme activity which is measured by supplying pineal homogenate by radioactive substrate precursors and by measuring the products that were produced, right? Over here exists the two terms, the samples taken by noon, right, and a samples taken by midnight, right, where where they are given a data, right, where you can find how all these substances were given, right? And now how the statistics work for this type of substance. By this level, take a look right at the graph, which is given by the statistics of those molecules, right, where the human chorionic gonad dropping, right, with respect of the week of pregnancies, right, is given, and how the interchangeability, right, of hormones in vertebrate species. You don't have to go through this, but if it's given, sometimes it's nice to know a little bit more. Rhythms in studying of this endocrinology, part of the experiments, making environments, right, manipulating. This involve the temperature in the environment, probably the light, which is called the light-dark cycle. The environmental parameters that never change has part of an experiment also. And these conditions are in which these experiments were housed, right? These hormones are found to have daily sensorial cycles or rhythms, which are called fluctuation. Rhythms implies one or more fluctuation repeated cycles. Interpreting the quantity of the hormones in a sample, and when this is necessary, we must know at what time of the day the sample was taken. The stage of the reproductive cycle, the sample was removed, and what month of the year the sample was acquired, right, and where the highest value, and with respect to the cycle, it was peak of maximum, and which one was the lowest value for that cycle, right? And the duration of light is considered 24 hours light. The dark cycle is called the photoperiod, right? A common period used with rats, it was designed by a specific team, right? They have their cycles, right? 14 hours of light and alterations with 10 hours of dark, right? And you know, take a look at the species specificity. The hormones are less specific. They are called species specific. Like in most organisms which are found small molecules, steroids, amine hormones, with very simple hormones or structures. The steroid testosterone has the same structure in all species. The rat testosterone is identified to human testosterone. testosterone obtained from rats will act in humans just like human testosterone. Other hormones are more species specific, often polypeptides or proteins. These hormones are different in structure and biological activity from one to other species. Because the sequence will be different in amino acids between growth hormone from whales and growth hormones from humans. So growth hormone of whales, for example, does not stimulate growth in humans. Monkeys' growth hormones, on the other hand, probably might be similar to the growth hormones of humans and can stimulate growth in humans. Some of the hierarchies, endocrine hierarchies, can be organized by function and interaction. And by this, the method of classifying hormones, the hormones are assigned to groups by their participation in regulation, making the classification a set of glands and hormones are listed in functional sequences or hierarchies. The generalized hierarchy model, brain regions, and the pineal gland regulates the hypothalamus. The hypothalamus secretes hormones that control the pituitary gland. Other hormones secreted in turn by pituitary gland takes control the gonads and the adrenal glands and the thyroid glands. The hierarchical endocrine arrangements gives a way to produce a regulatory tool of a powerful scale. A tiny signal is perceived by the brain where synthesis or release of just a few molecules convert to enormous effect. And because it's multiplying the strength of the signal and amplification, right, of that step. Amplification in endocrinology occurs in three independent ways: in the hierarchies, in the steroid mechanisms of actions, and the cyclic AMP mechanism of action. And why it is important to have an endocrine system carrying chemical messages if you already have a good nervous system, right? There are two possible reasons. First, the endocrine response is much slower than the speed of neural responses, uses less as 24 hours. And second, the hormone make use of the circulatory system to send the same signal to many parts of the body almost at the same time. To set point regulation, the neurosecretory cells in the hypothalamus might themselves be sensitive to various types of signals in the hypothalamus. Or the scientists have shown some cells directly sensitive to the temperature, circulating levels of glucose, salt concentrations in the blood, where these sensitive cells have been found set point regulations where has been proposed. The idea is that these cell groups work like thermostats to achieve a desired level of something, such as temperature, right? Uh, it can be increased or decreased. The cells are sensitive to increase or decrease in temperature in their immediate environment. The temperature of the blood bathing the cells is too high. The cells set off an endocrine sequence through the thyroid hierarchy, whose end results lower the body temperature. And therefore, on the other hand, if the temperature is very low, cells trigger endocrine events, raising body temperatures. Set the point regulations involved part of the regulations for thyroid blood temperature detection by a thermostat, growth hormone blood glucose detection by a glucostat, and for antidiuretic hormones blood salt concentration detected by an osmostat. Right. Take a look in your book, the tables, the general topics available, right? Applicable to endocrine cells, such as history, right? The hormones, function, regulation, the history. Talking in regards to negative feedback regulation, common in regulatory, right, for glands and target to hormones. Negative feedbacks is called servo mechanism. Hormones control their own concentration. Signal triggers secretion of hormones. When hormones reaches high levels, it turns off the cell that secretes it. Endocrinology negative feedback mechanisms are sometimes classified as long loop or short loop. A short loop feedback mechanism where the hormone feeds back on the gland that stimulates its production. Right. For example, growth hormone from anterior pituitary turns off the secretion of growth hormone, right? And release hormone in the hypothalamus. In long loop feedback mechanism, a hormone feeds further back on gland still higher in its hierarchy, such as thyroid hormone, right? Feeds back on hypothalamus, which secretes thyroid stimulation hormone, releasing hormones. Targets and receptors. The organs, tissue, or cells which hormones acts is called target. The uterus is an organ that is targeted by the hormone estrogen. More than one target can respond to more than one hormone. Receptors, target cells, used to recognize hormones found in cell membranes or cytoplasm. Targeting cells, the receptors are said to have specificity for hormones they recognize. Factors increasing or decreasing production or secretion of hormones from its gland is no regulation of hormones. Using a target organ, endocrinologists can determine the amount of response to a series of increasing doses of hormones. We know that performing a graph of the result called a dose response curve, right, which we learned, right, we can find how it works, right? And once a large dose gives to produce a response, is considered to have reached the threshold to be in the physiological range. It is known also as response of target cells proportional to the dose of the hormone. Hormones in the blood might be free or bound to other molecules, like we were saying, right? And the free form is believed responsible for the hormone's activity because the response is proportional to the dose. Like we were saying, the physiological range can be used to make standard curves for bioassays or hormone amounts. These amounts can be found normally in vivo, and they are called physiological concentrations, right? And large doses of hormones might produce one or three abnormal or pharmacological responses. First, the target might respond with a larger than normal response or a super response to a high dose of hormones. Second, the response might level off to a plateau, so that higher doses of hormone do not produce more than the maximum response of the target. And the third, high doses of hormone beyond the physiological range might actually inhibit the response of target. And the hormones can act in a variety of ways. They can stimulate a response of the target. They can inhibit something the target is doing, and where two hormones can oppose or antagonize the action of each other, and two or more hormones can act in concert upon a single target. In whichever case, they are called synergize each other. The mechanism of action for endocrinologists explain the way the hormone acts in the terms of chemical reactions, and they are mechanisms of actions. The mechanisms of actions fall into main categories: type of mechanisms for steroids and thyroid hormones, in which the action for proteins and amino hormones, right, which are non-roid or mechanisms of action. The semasiology, right, is a challenge, the beginning, right? It is challenging to give more than one term, right, at this level, and abbreviations for a single hormone. But it's necessary to learn hormones' names and spellings, but also which they are alliated, right? The hormones such as thyroid gland is called thyroid hormone, aka thyroxine, or it can be abbreviated T4, right? Some glands are listed in dictionaries only as adjectives. Pineal gland, thyroid gland, pituitary gland, and while others are listed as nouns, ovary, pancreas, testes, but people don't usually write ovary gland. Most of the glands are commonly referred to as nouns. The pituitary, right? Remaining objective in some dictionaries. Words are capitalized in endocrinology, usually derived from name of a person. Receptor target cells must have ways of recognizing the hormones to which they respond. Radioactive forms of hormones are variable, demonstrating specific binding of hormones to their targets, and more radioactive tagged hormones was found in targets than in non-targets. The specificity is because the receptor possessed by the target organ. The receptor accounts for the binding of hormones to target tissue. The receptors are proteins. The receptors are thought of having several binding sites, more than one per receptor. Molecules, hormones, binding to them are called ligands. Molecules compete for binding sites. Any kind of molecule binding to a receptor can compete. The fewer different kinds of molecules possibly binding, the more specific the binding is said to be. The affinity is used in chemistry, referring to the force by which atoms of certain elements combine and are remaining combined. One.